So let's just check out some of these ideas from chapter 3. So I just wanted to put a picture here of the phospholipid bilayer. This is the layer around every single one of your cells. In fact, this is the layer, a layer very similar to this one is around every single cell, whether it is prokaryotic or eukaryotic. Remember, prokaryotic cells still have a cell membrane, but they don't have membrane-bound organelles. You see there's these integral proteins and peripheral proteins, and these are going to be doing some stuff that we're going to talk about very shortly here. It also references here the fluid mosaic model of the plasma membrane. And basically what's happening is these proteins and things, they seem like they're fixed into place, but in reality they aren't. They're actually, they're able to move around and they sort of float through this fluid, and that's why it's called the fluid mosaic model of the plasma membrane. And the experiments they did to determine this were pretty cool. They like painted fluorescently on part of a plasma membrane, and then they were able to watch as the different components that they had painted were moving around. But yeah, that's the fluid mosaic model. This is a plasma membrane. Remember, these are hydrophilic. These phosphate heads love water, and these fatty acid tails, they're lipids, so they are hydrophobic. One question on the exam was, why is it that charged particles can't get through here? And it's because that they are too charged. So if you have something that is hydrophobic, like these fatty acid tails, it's not going to like things that are charged, and it's not going to allow them to get through. You also will have things that are too large that aren't able to get through because they're too large. But the question asks specifically, why can't charged particles get through? And that's the reason. Too much charge. Big theme in chapter 3 is the different types of transport that go on. Remember, we talked quite a bit about diffusion and osmosis, and you guys had that lab about it. But diffusion is what is referred to as passive transport. It does not require any energy. It just says, hey, if you have a whole bunch of particles all nearby each other, they are going to move away from each other until they are in equilibrium. And so diffusion is passive transport because it doesn't require any energy input from the cell. Things are just going to do it regardless of what the cell is up to. It's just going to happen passively. And you can see these particles can indeed get through your plasma membrane, your semi-permeable membrane, and so they are just going to do that. So that is diffusion. It is passive transport. This is exam 2 stuff, by the way. Diffusion is passive transport because it doesn't require any energy. So here's one that folks struggled with on the exam, and this is having to do with osmosis. So remember, I kept saying wherever solute goes, water is sure to follow. And this semi-permeable membrane here prevents your solute from moving from this area of high concentration to this area of low concentration, which would be diffusion. If this little red line wasn't here, these dots are going to move to the left side. But this line is here, so just like your cell membrane or like your dialysis bag, these solute particles can't actually move across. And so what happens is water is going to move toward that region of high concentration. So that is osmosis. It is the second type of passive transport, so where we have diffusion and osmosis. And we've talked sort of extensively about diffusion and osmosis and the various tonicities of solutions. So yeah, hypertonic, where is the solute? There's a bunch of solute in the solution, right? And so water is leaving those cells. Isotonic, it's the same concentration inside and out, and so water is, it may be exiting sometimes, maybe entering sometimes, but there's no net exchange. But I shouldn't say it like that. There's no net movement in either direction. So they're not filling up, they're not becoming deflated. And then when you have a hypotonic solution, there's more solution inside, or more solute inside of your cells. And so wherever solute goes, water is sure to follow, and so it's going to go into the cells, causing them to explode. So if you put cells in a hypotonic solution, they're going to fill with water and occasionally explode. Same principle here for plant cells, but they won't explode because they have cell walls. Okay, so this is a transmembrane protein. It's a transmembrane protein because it is spanning this membrane right here. Okay, and what's happening here is you have a high concentration of these positive particles here on the inside. And so what's happening is these positive particles are getting pushed out through this channel because there is such a high concentration of them. Okay, and we talked about this in the electron transport chain. And in the electron transport chain, you actually have an active process pumping those things out. But here, this is more like a facilitated diffusion is what it's called. So your first type of what's referred to as, your third type of passive transport, because no energy required, is facilitated diffusion. It's facilitated because there's a channel for it, and it's diffusion because all of this high concentration of positive charges are getting pushed out through it. Okay, so our first type of active transport is the sodium-potassium pump. The potassium pump winds up moving around positive charges, but it's going to pump different numbers of positive charges. And so let's say it pumps out 3 sodium ions, it's only going to pump in 2 potassium ions. And so you don't need to worry about the mechanics of that necessarily, but you should know that the sodium-potassium pump is building an electrochemical gradient. It is building a difference in charge. When I say electrochemical gradient, I just mean a difference in charge. It's building a difference in charge across this plasma membrane. Okay, and you can see here this ATP molecule is being used up in the process. So this is active transport because it requires energy. And then we have 3 other types of active transport that are somewhat related to one another. Here we have phagocytosis, where a large particle is being sort of enveloped or invaginated into a cell. So the large particle is going to come and land here, and the cell is going to sort of move around it until it gets put into this vacuole thing, and the cell has it. The next kind of active transport is penocytosis. This is just another variation, again, of endocytosis. So phagocytosis and receptor-mediated endocytosis are all endocytosis, and they're all active transport. And in penocytosis, the cell is bringing in water, basically. Just little tiny bits of fluid that it's bringing in through a similar process as phagocytosis. Okay, just works the same way. Water hits the cell, and it sort of folds over and brings in whatever hit it into this little vacuole here. We also have receptor-mediated endocytosis, and in this type, don't worry about this term clathrin right here. That's not necessarily important. But what happens is, well, it's not important right now anyway, but we have these receptors, we have these things on the outside of the cell that are looking for stuff. In this case, they're looking for stars, whatever stars happen to be. And whenever a star lands on one of these receptors, that's what causes the cell to fold up and bring it in to one of these vesicles. Okay, so that is receptor-mediated endocytosis. Okay, these are just different forms of endocytosis. They are active. Okay, these require energy. So here we have like the opposite process, which is exocytosis. Okay, and you know, endocytosis kind of gives you a clue for what's happening. Things are going into the cell. Exo, things are exiting the cell. And so what happens is this vesicle comes up and floats, and it fuses with the plasma membrane. Okay, it actually comes up, it's got whatever your cell is trying to get rid of in it, and it actually physically fuses to the plasma membrane and sort of unfolds and opens up and lets whatever it has inside of it go. Okay, so exocytosis, this is another form of active transport, and It is taking stuff from inside your cell, putting it into a vesicle, sending that vesicle to the edge, to the plasma membrane, having that vesicle fuse with the plasma membrane, and letting all that stuff go. So that's the last kind of transport that I'm going to talk about in this lecture. So again, diffusion and osmosis, as well as facilitated diffusion, are passive transport. They do not require energy. Then we have endocytosis, which includes pinocytosis, phagocytosis, and receptor-mediated endocytosis. Okay. Those are active transport, as is the ion pumps that you saw before, as is exocytosis, which we just talked about.